Welcome to my nano talk on mRNA vaccines. mRNA vaccines are not new. They are used for 100 years. The vaccines against measles, mumps, yellow fever, rubella all consist of mRNA viruses that are attenuated. When injected, these vaccines deliver their messenger RNA in the host cells. The host cells produce the proteins of the viruses and mount an immune response. What is new is a synthetic mRNA vaccine where the messenger RNA is produced by in vitro transcription and is encapsulated in a lipid bilayer consisting of 4 lipids. The mode of action of a synthetic mRNA vaccine is the same than the mode of action of a natural mRNA vaccine used since 100 years. mRNA is pictured here. This is a cell, a mammalian cell with a nucleus. In the nucleus, you have a DNA. Part of the DNA is transcribed into pre-messenger mRNA. In the nucleus, the pre-messenger mRNA is matured. It's capped at the 5' end with this structure added at the 5' end, and it's polyadenylated at the 3' end. This capped polyadenylated mature messenger RNA goes in the cytosol, where it's further modified and translated into a protein. This messenger RNA in the cytosol is transient. It's being there expressed in protein for a few minutes till a few hours. In vitro, it's possible to produce this mature messenger RNA. It comes in 2 flavors, either non-modified or modified. When it's non-modified, it's produced with the 4 canonical nucleotides ACGU. And this non-modified mRNA in the body is recognized by receptors in immune cells. These receptors, called Toll-like receptors, stimulate inflammation when they detect nucleic acids, for example, non-modified mRNA that is injected. So when you inject this non-modified mRNA, because of inflammation, you can do vaccination. And this is used and developed as anti-cancer vaccination, anti-allergy vaccination, and has been approved recently against COVID-19 as antivirus vaccination. Another way to produce mRNA is to modify the mRNA with pseudouridine. And this modification allows the mRNA to escape the recognition by Toll-like receptors. So this modified mRNA is not immunostimulating. You can inject it and do not trigger an immune response. That's why it's used for gene therapy. For example, for expression of therapeutic proteins like erythropoietin or antibodies, it can be used with modified RNA for cellular reprogramming or genome engineering, for example, with Cas9. Recently, in 2017, it was published that surprisingly, this modified non-immunogenic mRNA can also be used for vaccination when it's given intradermal or intramuscular. But so far, there is no evidence whether modified mRNA is better or less good than non-modified mRNA for vaccination. And both are used. This is how mRNA is produced in vitro. You need a few reagents. First, a plasmid. This is a DNA molecule made by molecular engineering and extracted from the bacteria. It's linearized with a restriction enzyme, and the linear plasmid DNA is transcribed using an RNA polymerase, nucleotides, and the cap analog at the 5' into mature messenger mRNA. After this transcription step, you add the DNAs to destroy the DNA strands, and then the mRNA is recovered by selective precipitation with lithium chloride or more advanced method to have higher grade of messenger mRNA. Each of these steps is robust, which means that linearization, transcription, DNA treatment always work, whatever is the starting plasmid, whatever is the sequence in the starting plasmid, whether it codes for a virus protein or for a cancer protein or for allergy protein. This process is always the same. It works always. Within a few hours, through the steps, you get mRNA. This is in the laboratory. In pharmaceutical production, it takes a bit more time because you need in-process control and end product controls on the mRNA. A few facts on synthetic mRNA. First, mRNA is very stable. Contrarily to what people think, mRNA is the most stable biological molecule. It's the only biological molecule that you can heat up to 95 degrees, lyophilize, freeze, and that doesn't lose its activity. If you heat up to 95 degrees DNA or proteins or viruses, they will be deactivated. It's not the case for mRNA. So mRNA is the most stable biological molecule. On the contrary, the formulation of RNA, the pharmaceutical formulation of RNA in liposome may not be stable. It may aggregate, it may change size, form over the time, or by freeze drying. So that's why the formulation of the mRNA vaccines have to be stored at a low temperature. It's because of the liposome, not because of the mRNA. RNA in liposome is already approved as a drug. It's called Onpattro. It was approved in 2018. It's a drug used to treat patients suffering from hereditary transthyretin-mediated amyloidosis. And these patients receive 30 milligrams of the RNA intravenous every 3 weeks. It's not a messenger RNA, it's a siRNA. So this treatment exists, and as you will see, the vaccine uses 30 micrograms, 1,000 times less RNA than in Onpattro. Fourth point, IVT mRNA vaccines are vegan. As I showed you on the previous slide, the production requires enzymes which come from bacteria and chemicals like nucleotides. So the production is vegan. Finally, the production of mRNA doesn't require big infrastructures. 1 million of doses of mRNA vaccine can be produced with 6 liters in vitro transcription. In comparison, when you produce viruses for vaccination, you need a few thousand liters of cell culture in order to extract enough viruses for 1 million of doses. A few words on the timelines of IVT synthetic mRNA vaccines. The first demonstration that in vitro transcribed mRNA can be used in vivo was published in 1990 by an American team who has shown in mice that in vitro transcribed mRNA coding for beta-galactosidase injected naked in the muscle is expressed into beta-galactosidase. 3 years later, a French team has shown that the mRNA coding for influenza protein and packaged in liposomes can vaccinate mice. This was the first disclosure of mRNA vaccine, and it was mRNA in liposome, very similar to the COVID vaccines that have been approved. Later on, some more teams have shown that naked mRNA or other form of packaging can be used to vaccinate mice. And my colleague Ingmar Her, who has done his work under the guidance of Professor Ramondse and Professor Gunther Jung, has published in 2000 this article, and we have created CureVac in order to develop the mRNA vaccines and bring it to the clinic. One of the goals of CureVac was, of course, to improve the mRNA vaccine and to produce mRNA for the clinic in so-called GMP production, good manufacturing practices. We could produce then mRNA to inject in people, and it was first injected into me to check that naked mRNA could also be taken up into the human skin. Here is my leg. I got a few injections of mRNA coding for luciferase, and a few hours after injection, under local anesthesia, we made punch biopsies here, and we measured in vitro whether the punch biopsies were expressing light, and that was the case. So these experiments published in 2007 have shown that when you inject in human skin naked mRNA, you find the same result than in mouse skin. That means the mRNA is taken up and translated into protein, in this case, firefly protein. Now for the COVID-19 vaccines, the three big companies have been working since the beginning of 2020 on the vaccines, and they came up all with the mRNA vaccine in liposomes that is given intramuscular and that is coding for the spike of SARS-CoV-2. First to start was Moderna, who is using modified mRNA. The name Moderna means modified mRNA. They use only modified mRNA, and they have injected the first healthy volunteers on March 16. They had 3 doses in their dose escalation phase 1 study from 25 to 250 micrograms. BioNTech started 1 month later with the first injection on the 25th of April, and BioNTech had 4 different vaccines in the test in April, and it included non-modified as well as modified mRNA vaccines. And the dose range was from 1 to 100 micrograms. CureVac has started later with a dose range between 2 micrograms and 12 micrograms of non-modified mRNA. The phase 1 studies are published, and they have shown that this formulation of mRNA in liposome at these different dosage is safe and well tolerated, although there were some side effects, which is expected for a strong vaccine, inducing a strong immune response. Importantly, all volunteers seroconverted, even at the very low dose of the vaccine. And they were neutralizing antibodies after the boost at the doses used for the phase 3 study. So, and for the registration, which is 100 microgram of mRNA for Moderna and 30 microgram of mRNA for BioNTech. CureVac is ongoing in its phase 3 study with 12 microgram of non-modified mRNA. The phase 3 studies from BioNTech and Moderna have shown 95% protection against COVID-19. And more recently in Israel, that have massively vaccinated its population, it was demonstrated that the BioNTech Pfizer vaccine gives a 94% protection against infection by SARS-CoV-2. Here is an example of the antibody response triggered by these vaccines. Here, the BioNTech Pfizer vaccine given in healthy volunteers. Some volunteers here got a placebo, and they don't have in their serum any neutralizing antibodies against SARS-CoV-2. These are sera from volunteers who have recovered from the COVID-19, and they have neutralizing antibodies against SARS-CoV-2. These here are the healthy volunteers who got the vaccine at different dose, 10 to 30 microgram, younger people or elderly people. You see that in all the cases at day 28, which is 1 week after the second injection, all volunteers have neutralizing antibodies against SARS-CoV-2, even at the low dose of 10 microgram, but even higher at the 30 microgram, whether they are young or whether they are old. So this shows how strong is the mRNA vaccine, even at this low dose in both young and elderly population. With that, I'm finished. You can find more detail about the mRNA vaccine in my recent review published in February 2021, and I'm looking forward for your questions.
Thank you very much, Mr. Pascolo, for your talk. We have already received several questions, and the first question we have received concerns the difference between modified and non-modified mRNA. And the question is, does modified or non-modified mRNA need an adjuvant for activating the immune system? And are modified and non-modified mRNA equally good in stimulating our immune system? We have already received several questions.
Do you hear me well? So whether the modification is needed or whether it increases or decreases the efficacy of mRNA, we don't know. It was never compared exactly non-modified and modified mRNA in vaccines. So BioNTech has done it. In April, they had exactly the same sequence of mRNA in the exact same formulation, in the exact same site of injection, modified versus non-modified. They didn't yet publish the data they had with a non-modified. They published the data they had with a modified, but not with a non-modified. So as soon as they will publish the data they had with a non-modified, we will see in humans the exact same sequence of mRNA in the exact same formulation, in the exact same site of injection, whether there is a difference in the side effects and in the efficacy. So far, there was no such precise exact comparison between modified and non-modified in vaccines. So both work, and non-modified has been used more, especially against cancer, than modified. But in the context of COVID, we don't know, and prophylactic vaccines, we don't know whether the modification is good or bad. In the non-modified mRNA, because it's not anymore a toll-like receptor agonist, it's highly probable, it was also kind of shown, that the adjuvant effect comes from the lipid, from the liposome itself, is inflammatory, So when you inject the non-modified mRNA in the liposome, the inflammation probably, highly probably, comes from the liposome, also from the damage made to the muscle by injection, but most probably, and data are supporting that, from the liposome itself.
Thank you very much. We already have several following questions. And one is, you mentioned that different companies have different dosages for the vaccines. And the question is, how does it come that the dosages are so different from company to company?
They're not that different. I mean, there are 100 micrograms for Moderna to 12 micrograms for CureVac. But if you remember the graphic I showed you from BioNTech, the vaccine worked with 10 micrograms, with 20 micrograms, and with 30 micrograms. So especially in healthy younger volunteers. So BioNTech could have probably come also with a 10 microgram vaccine, it would have worked. So they choose 30 microgram, they could have chosen more or less. The point is that you want to have a dose that will work in really nearly 100% of the people. So even if a dose of 10 micrograms from the BioNTech vaccine would have worked in all younger healthy people, you are not sure that somebody immunosuppressed or weak would respond. So companies have probably chosen a dose that is higher than the minimum dose. BioNTech would have probably worked with 30 microgram instead of 100, and Moderna would have worked probably, they would have used 30 microgram instead of 100. In their phase 1 study, the 25 microgram dose of Moderna was good. It induced immune response neutralizing antibodies in all participants, healthy volunteers. So, and again, BioNTech Pfizer also worked at 10 microgram in all healthy volunteers. So the doses was, all doses worked, basically, what they tested. So they choose a dose that they think was good, safe, and still would cover everyone, but they could have chosen different doses. So at the end, I think that all vaccines would work at 12 microgram, like the one of CureVac. So if Moderna or BioNTech Pfizer would be 12 microgram, they would also work. So they choose 30 microgram to be a little bit secure that there will be no one who will be resistant to the vaccine. And sorry, there is also small differences in the liposomes. It's not the same liposome in Moderna, BioNTech, and CureVac. So it's also possible that the liposome of one company is better than the other company. So it could make a small difference. And of course, there is a difference between the modified and the non-modified mRNA. So maybe the non-modified is better than the modified, but these are probably small aspects.
Before we go to our next question, there is a very short question which you could maybe answer, and it is whether the BioNTech Pfizer now is using modified or non-modified mRNA in the available vaccine.
The BioNTech Pfizer is using modified mRNA. They have tested non-modified and modified, and they have chosen modified for the phase 3, but we don't know why, because they didn't publish the results they had in phase 1 with the non-modified vaccine. But the registration for the BioNTech Pfizer is the modified version of the mRNA. For Moderna also is modified. And for CureVac, it's non-modified. It's not yet registered. We are hoping that they will get good results.
Another question some people asked is that you mentioned that it is actually quite easy to produce the mRNA vaccines, and now we have several delivery problems, and people are asking, why do we have all these delivery problems by the companies now if it is actually that easy to produce the mRNA vaccine?
What I've showed is that it's very easy to produce mRNA. So the mRNA is very easy and fast to produce. What is a little bit more demanding is the lipid layer around. So the formulation, this requires 4 lipids. The 4 lipids, the lipid chemistry is difficult to make lipids, to purify lipids is much more difficult than to make and purify mRNA. So the struggle is to get the 4 lipids that you need for the liposomes of Moderna, BioNTech, or CureVac, and to get the providers delivering you this lipid, and then to mix the mRNA with this lipid. It also requires some special installations. So the bottleneck is not the mRNA, it's the liposome. And that's why companies, Moderna, BioNTech, and also CureVac, are working on new nanoparticles, which will not rely on these liposomes, which will rely on other liposomes, easier to make with, for example, only 1 lipid, or on nanoparticles which require polyplexes, in polycations, for example, polymers, which are easier to produce in large amount than lipids. So the tendency now is to replace this liposomal formulation of mRNA by other formulations, because mRNA is easy to have. It's the liposomal formulation which is difficult to get and to make. And then it's the liposomal formulation which is unstable, which has to be stored at low temperature. So if you have a new formulation of mRNA, if you have naked mRNA or mRNA in an easy formulation, then it could be much easier to do and stable at room temperature. So the issue is really much more the lipid bilayer around the mRNA than the mRNA itself. The mRNA itself is easy to produce.
We have our next bit more general question, and this is whether all vaccines are preventing an infection with the virus, or do they just prevent the severe symptoms, or do they really prevent an outbreak of the disease?
The endpoints of the clinical studies were to prevent the disease, because you can measure the disease. So you can see in your placebo or in your healthy volunteers vaccinated, you can see how many have symptoms of the disease. So the endpoints of the clinical studies were really to see how many people suffer from COVID in the placebo versus vaccinated people. It's much harder to test the infectivity, so under placebo or vaccinated. But now it came from the Israel data. So in Israel, that has covered this population, they could see the rate of infection, so non-symptomatic infection, just PCR positive people without symptoms. They could see how many people got PCR positive in the vaccinated people versus the non-vaccinated people, and they could confirm that there is 95% protection against infection. So not only against the disease it was shown in the clinical study from BioNTech, Pfizer, and Moderna, but now in Israel, we also know that there is 94% protection against the infection. Which still means, that there is 5 to 10% possibility when you are vaccinated to still get COVID and to still get infected. So people should not believe that there is 100% protection. So we hear sometimes people saying, yeah, I know someone who was vaccinated, he got COVID. Yes, it's 90 to 95% protection. So you still have people who get vaccinated twice with the BioNTech, Pfizer, or Moderna, and who still get signs of COVID, who still get positive in PCR, because it's not 100%, it's 90 to 95%.
So it actually means that you still have to be quite careful, because it's not a really 100% protection.
Exactly.
Then we also have a question. So you mentioned that there are mRNA and vector vaccines, and we have a question whether there are also other vaccine techniques than vector and mRNA, and then what are the differences between the vector mRNA, and if there also exist other techniques?
Against COVID, there are 4 platforms that have been validated in the world: protein-based vaccine, mRNA-based vaccines, adenoviruses-based vaccine, and inactivated SARS-CoV-2-based vaccines. Inactivated SARS-CoV-2 has been mostly done in China, and we don't have it here in Switzerland. Protein-based vaccines is the Novavax. It has been approved in several countries, not yet in Switzerland, although Switzerland has reserved it, and it looks good, the data of protection of the Novavax vaccine, protein-based. The adenovirus-based vaccine, Johnson & Johnson, AstraZeneca, Sputnik V. So the AstraZeneca has been reserved in Switzerland. It's not yet approved, but it's adenoviruses. So yes, there are these 5 different platforms: protein, adeno, mRNA, inactivated virus, 4 platforms that have all been used in the world, all been validated, being quite good. I must say that the efficacy data are best with the mRNA vaccines that protect with 95%, that provide 95% protection. Other vaccines like the AstraZeneca provide less protection. And also the mRNA vaccines, the Moderna and the BioNTech, Pfizer, protect against all variants, while the AstraZeneca, the adenovirus, the recombinant adenovirus, does not protect against the South African variant. So the mRNA vaccine seems to be giving the better protection and the broader protection.
We already have a next question concerning the current development of mRNA vaccine. And the question is whether there are technologies under development that reduce the large amounts of injected mRNA, which is necessary for a potent immune response.
What do you mean? So higher amount, or?
The question is probably whether there are technologies with which you can reduce the amount of mRNA that is needed.
All companies are working in getting immune response with less mRNA, because of course, if you take the case of Moderna with 100 micrograms per dose, if they manage to have a new formulation, the lipid or the liposome or the nanoparticle, that allows you to get 10 times better efficacy, then that means you need only 10 micrograms in your vaccine, which means with your stock of vaccine with 1 kilogram, for example, instead of doing 1 million doses, you will do 10 million doses. So all companies are working in improving the mRNA vaccine. In the last 20 years, we have improved it so much. 20 years ago in the first clinical studies, we injected a few hundred micrograms every week to patients to get a good immune response. Now we inject twice 30 micrograms. So in 20 years, it has progress enormously, and with science it will further progress. In 2 years probably we will have mRNA vaccines with only 1 or 2 micrograms in it. In 10 years maybe with 0.1 microgram. So the improvement of the mRNA, of the formulation of the mRNA, will allow to continue to improve the mRNA vaccine, which means to make it even more efficacious, which means that you will get even lower dose to have a high immune response. That's constantly ongoing. It's not the end of the mRNA vaccine, it's the beginning. And it will be constantly improved, which will mean reducing the dose that is needed in order to get a high immune response.
Thank you very much. We have another question which concerns whether you're going to be positive in a PCR test shortly after your vaccination, or is it the case that the test targets other gene sequences?
The test targets few gene sequences in the SARS-CoV-2, so you will not get positive if you get injected with mRNA coding for the spike. Also, it will stay anyway in the locally in your muscle of the arm, so it will not go in the nose. So no, you will not get PCR positive after getting injection of the mRNA vaccines.
Yeah. This sounds great. And maybe for our last question, maybe you can tell us whether there is already information on how the synthetic mRNA can be used as a cancer treatment.
It has been the focus of CureVac at the beginning and also of BioNTech to do mRNA vaccines against cancer. We know that it's hard to make vaccines against cancer on any platform, peptide, proteins. It's the same for HIV. So there have been a lot of vaccines that have triggered immune response against cancer or against HIV. But clinically, although you get an immune response, you don't get a control of the disease, HIV or cancer. So the mRNA vaccine have fallen under this data. So they induced the mRNA vaccines against cancer that have been tested have induced immunity against cancer. So that was successful. But this immune response, like in cases of other vaccines, did not efficiently control cancer growth. So now the mRNA vaccines are being developed to target mutations, and the flexibility of the production of mRNA vaccine allows to have individualized vaccines. So what is ongoing now mostly is to sequence the tumors of patients, of cancer patients. The sequence, once it's known, you find the mutations in the tumor versus healthy tissues, and you can generate mRNA coding for this mutation and vaccinate each individual with its own mRNA. It's fully individualized that codes for its mutation. BioNTech does that with Genentech. They're in phase 2. So each patient receives its own mRNA vaccines coding for its own mutation. And from the biopsy till the mRNA vaccine is ready for one person, it's 3 months. So the flexibility of the production of mRNA allows this technology, which is new, to really target in a totally individualized way the mutation in each cancer. So in theory, it can treat any type of cancer, melanoma, lung cancer, etc., because you target the mutation that each patient has in its own cancer. So this is one development of the mRNA vaccine. It's allowed by the flexibility of the production of the mRNA vaccine, as we have seen in the pandemic. It takes only a few weeks to develop an mRNA vaccine. It's easy, and so it goes fast. So that's why cancer patients can get this new therapy. And this is combined also with some standard immunotherapy, radiotherapy, and chemotherapy to have a double effect on the cancer vaccination plus other type of therapies to synergize and really get rid of the cancer and prevent relapses.
Yes, this sounds very promising if we can also use the mRNA for cancer treatment. And I thank you very much that you were here this evening, Mr. Pascolo, and answered our viewers' questions. And I hand now over to Ophelie for our second talk.
Thank you for the
very much,
invitation.
Mr.
Good
Pascolo.
evening. Thank you.
Thank you